Good morning, everybody. I would like to thank the organizing committee for inviting me, and I'm excited to tell you about the new work we're doing on phage-bacteria interactions. As you know, with the microbiome, what we're trying to capture is really the interactions of what is happening. Who is there? What is it doing? How does it react? And when does it really matter? In the work we do in my lab, we're very interested in the effect of pathogens on the disruption of the airway. And we've published a few papers that describe some of these studies. In one recent study, we actually looked at the interaction of fungus and bacteria and how these interactions can impact the airways. But one player that's important when we consider these interactions and the dynamics of the microbiome are phages. Now, if you know anything about phages, is that they can have a very dynamic effect on their host. Phages can be beneficial to their host, shuttling genes that confer some selective advantage. They can also kill their host and promote competitors in the microbiome. And they're also important in capturing bacteria that will grow better with phages, or you can see expansion of phage populations. One aspect that is less often reported is the fact that phages can interact directly with the immune system. And I won't go into that specifically today, but it is interesting to see that they can interface with macrophages directly and in this way indirectly influence the immune cell activity. And in our studies, what we focus on is really the disruption of the upper airways and consequently what can be the lower airways with viruses like influenza and more recently SARS-CoV-2. There are commensals or normal members of the microbiome in the respiratory tract that can become pathogenic because of the lack of competition as other microbes are disrupted because of the viruses. And it's that aspect that we try to capture when we look at the microbiome in the respiratory tract during viral infection. We've done a study with Elodie Ghedin that is specifically on looking at influenza transmission within households. And these are very nice data sets to work on because we have a lot of information on the subjects on what antibiotics they take, how they interact with each other. And that's what's nice about having an influenza household transmission study. And we published a study earlier this year where we characterize antibiotic resistance genes using the metatranscriptome data. Now, this was a pilot study, and what we really wanted to do was to better understand transmission and whether influenza could lead to a higher transmission of antibiotic resistance genes or bacteria between individuals within a household. Because the main question we have is influenza disrupting the microbiome in such a way that it can even lead to transmission of pathogenic bacteria during a flu infection. To do this, we had access through Aubrey to even more samples within different transmission households. And what we call different transmission households is when you have an index case that has transmitted to every member of the household or to just a few members. And so we collected 10 different households, samples from all the individuals within these households for the analyses that I will show you today. Now, while our question was specifically, do we see higher transmission of influenza? What I'm showing you today is more, can we capture some dynamics of interactions between the phage and bacteria in these different households? And can we see any differences? Now, this was a feat for the analyses. Lingdi Zhang, a fifth-year PhD student in my lab, did all these analyses. She processed over 200 samples for both the RNA and the DNA. Samples came from 54 subjects and were collected at different time points. So that gives us also the ability to do some dynamic analyses. And what we do is the expression data, so from the metatranscriptome, and looking at abundance, but also at other aspects of the phages and the bacteria specifically from that data. And that's what I want to discuss specifically with you today. Now, of course, the big question is, how is the microbial ecology impacted during flu infection? Looking at microbial community, pathways, antibiotic resistance, but what about this bacteria-phage interactions? Now, the way to do this in a more standard approach is to do what are called enrichment analyses, and I'll describe what that is, or also looking at co-occurrence of bacteria and phages. If one is absent, is the other present, or are they both present at the same time? And that's another approach to look at interactions. So what do I mean by enrichment? Here in our analyses specifically of these samples, we have a representation of the metagenome in orange, the metatranscriptome in blue. And what you have here is differential abundance or differential expression based on whether flu is present in the sample or flu is absent. And you have as a control right here in the metatranscriptome, you see this very long line that indicates influenza virus, which we would hope to capture in the metatranscriptome data. But if I focus a little bit on the taxa that we do pick up as being differentially abundant or differentially expressed, we see here Streptococcus, where at the abundance level, there is no difference between flu or no flu, but there is a difference in functionally how genes are expressed from these different species, with a flu impacting this expression. And then we capture two phages that are differentially abundant. Now, from that type of data, we would infer that there's probably an interaction with some of these strep species and strep phages. And we have, for example, one of these phages that is found at a high level in flu, while the other is present at a low level. And we can sort of infer the dynamics that happen there. But again, this is not a direct interaction. This is really inference from the enrichment data. Another way to look at these interactions is to look at co-occurrence networks. Now, a way to do this, here is an example of a study we published this year. It's not at all from flu data, it's from sewage data, work done by another PhD student in my lab, Kristen Gulino. And here, using a tool that was actually developed to capture microbe metabolite interaction, Rich Bono's lab at NYU decided to adapt it to phage and bacteria. And here what you can look at is whether any of the phages, represented as red triangles here, seem to be interacting with different types of bacteria. And if we focus on some of these interactions, you can see here on the left what you see with the triangle. You see crassphage, and crassphage is a newly discovered phage that is known to be present in everybody's gut. So we find it a lot in the gut microbiome, and it here co-occurs with bacteroides, which are known hosts of crassphage. And we can go into different sections of these networks and identify other phages. Here in this example, Lactococcus similarly is found in bacteria of the same phylum. So this is a way to do it also. But what I want to show you is how we can use the metagenomic data to look at direct interactions where we can link phages with their metagenome. So how can we do this? We can use the CRISPR system that is present in most bacterial species. Some species do not have a CRISPR system, but that system records the phage infection, because any virus that has infected the bacteria will have some of its DNA or even RNA that ends up integrated into a CRISPR array. And it's represented here by a spacer. And the spacers that here are red, purple, and yellow represent pieces of the viruses that have infected the bacteria at some point in its history. So we're using that information specifically from the bacteria to then track back to the bacteria and phages that we find in our samples. This is the analytical pipeline. I've put in square brackets any of the tools that we've used, and this can be done in different ways, and this is the way we've done it. So basically we take all the sequence reads at each time point for each individual, and we've actually pooled from the time points for each individual. And mine is filtered out with the human and ribosomal RNA reads. We then do an assignment of taxonomy using Kraken. So this is a tool that will tell you what is a bacterial read and give it a taxon, and what is a virus read. In this case, we've used it to assign our bacterial reads, and then we've assembled our bacterial reads into bacterial contigs to which we've assigned taxonomy. We've also used these bacterial reads to then run them through a tool called Metacrast that identifies these spacers in these CRISPR arrays. And we've used these spacers specifically to then map back to the bacterial contigs that we had and to map to the virus contigs that we've assembled in a slightly different way, but using a very similar approach to our bacterial reads. So now what we have is an active presence of these phages in our samples, and look at whether any of these phages are present within the bacterial arrays, indicating in a dynamic way that there has been interaction at some point within these individuals. And so what we get from that are more than 1,500 phage-host associations that we're able to capture across all our samples. When we talk about phage-host interactions and we are interested in the microbial ecology, we can say, you know, what is happening in the respiratory tract? And you can have different types of structures. You can have what's called a nested structure of interaction or a modular structure. And what does that mean? In the nested, you have a hierarchy of infection and resistance of these bacteria to their phages. And you have some phages that are generalists, so a phage can infect multiple bacteria species, or you can have a specialist, and those are rarer, where you have one specific phage that infects one specific bacteria. In the modular structure, it's where you'll have an ecological framework that is basically specific interactions of bacteria and phages, but that form their own units. And what we see in the respiratory tract is actually a combination of both. So we see a modular structure, but within these modules, we have a lot of generalists and specialists. So we have this internal nestedness. Now, what that means specifically in the case of a flu infection is unclear, but we are seeing this interesting ecology or microbial interactions with their phages within the airways. So if we look at our subjects or individuals that were either infected or not infected with flu, and we specifically look at our bacterial taxa that you see here on the y-axis, and you see phages on the x-axis, we can then now look at how many individuals appear to have these interactions. And the color code is such that if you're dark purple, that means that interaction is found in many of the individuals for which we did the analyses. If it's gray, that means we found that interaction only in one individual. And what that shows you is that in most cases, we have a lot of unique interactions that we were able to capture. Now, many of the ones where we find conserved across individuals are known bacteria such as Streptococcus and Prevotella and Neisseria that are known bacteria that we find in the upper airways. And so we are seeing them pretty conserved across individuals with known phages that infect them. We were interested in comparing flu and no flu, and this is how our samples are sort of taken. We take all the samples at all time points from the not infected individuals. So these are no flu households. We take only the infected time points from our infected households. And here I show the genus of the bacteria and just the genus of what would be the phages for these interactions on the X and Y axes. And then what I've highlighted in red are specific groups of bacteria where we do see changes between no infection and infection. And if you see that there's Moraxella, which is the fourth one that's highlighted, we see that in infection, Moraxella and the phages, we do find these interactions which we don't find in the no infection. And you do see differences in number of, or the percentage of samples where we find these interactions. So this is just, you know, we're still doing a lot of analyses on this, but this shows you how there's far more resolution when you use your CRISPR arrays than when you just use your enrichment method, and less, you know, complicated than when you use your network interactions that we see from the co-occurrence networks that we looked at. So from these analyses, what we've seen so far is that CRISPR arrays can be used to capture direct phage-bacteria interactions. And what we saw is that these interactions in the airways appear to indicate high diversity and high availability of resources of the bacteria and their phages. And what we are starting to see at a better resolution is the impact of influenza on the microbiome that we are detecting through these interactions. So I'd like to just acknowledge other people. I've mentioned Rich Bono, and there are other members of his team with whom we've worked on these interactions. And actually, Rich and I co-mentored Lingdi Zhang, who will be graduating in about 6 months. And Jahan Rahman is an undergraduate student who worked with Lingdi and with Kristen on the CRISPR arrays and identifying these interactions. I am now at the NIH, but I used to be at NYU until last May. And so most of the analyses were done, and that study was done mostly at NYU. Thank you very much.